Welcome to RadOnc Smart Review RadBio Edition.
And today, we're really digging into the absolute core wiring of cancer cells.
Yeah, we're talking about those cellular signaling pathways. They basically dictate when a cell grows, when it divides, or when it decides it's time to check out.
Exactly. And understanding how cancer hijacks these networks is just so crucial, not just for passing the boards, obviously, but for really getting why we use the treatments we do in the clinic.
That's exactly right. We're going to focus on some major players you'll see come up again and again. The epidermal growth factor receptor pathway.
Right, EGFR.
The cascade involving the RAS protein and mitogen-activated protein kinases. So the MAP pathway.
Mm-hmm.
The pathway centered on PI3 kinase and AKT.
Yeah.
And then those heavy hitter transcription factors. Myc and
We've talked
NF-kappaB.
before about oncogenes being like stuck accelerators and tumor suppressors as well, broken brakes.
Right.
Today, we're kind of lifting the hood to see the actual internal circuitry, how those accelerators and brakes are really connected and
when this wiring gets messed up, that's when cancer thrives. Critically, it really influences how tumors respond or don't respond to radiation and systemic therapies.
Okay, so let's start right at the edge of the cell, on the surface, with the epidermal growth factor receptor signaling.
EGFR. Think of this as one of the cell's main antennas, listening for those growth signals from the outside world.
Right, commonly called EGFR, but you'll also see it as ERBB1 or HER1. It's part of a whole family, the ERBB family of receptor tyrosine kinases.
So they sit there on the cell surface waiting to bind to ligands, these external signaling molecules like epidermal growth factor itself or transforming growth factor alpha.
And just a quick point here, TGF-alpha is different from TGF-beta. TGF-beta often has more of an inhibitory role on growth, so don't mix those up.
Good point. So how does binding that ligand on the outside actually, send a signal inside the cell? It's kind of a chain reaction.
It is. Step 1. The ligand binds. EGF or TGF-alpha latches onto the EGFR receptor outside the cell.
And this causes the receptor to change its shape, right?
Exactly. That shape changes the key. It triggers step 2, dimerization. The receptor pairs up.
It can team up with another EGFR that's a homodimer, or sometimes with a related receptor like HER2, forming a heterodimer.
And bringing those receptors together like that pulls their intracellular parts closer.
Those are the tyrosine kinase domains.
Right. And when they get close, step 3 happens.
Yeah.
Autophosphorylation. They get active and start adding phosphate groups onto specific tyrosine residues, either on themselves or on their partner. Transphosphorylation, sometimes called.
Okay, so now you've got these phosphorylated tyrosines sticking out inside the cell. What happens next?
Step 4, they become docking sites. They recruit adapter proteins, sort of like molecular connectors and other signaling enzymes.
These adapters often have special domains, SH2 domains, that specifically recognize those phosphotyrosines.
Yeah, a classic example is GRB2. That's an adapter protein that docks there, and then it recruits another player called SOS.
And recruiting those proteins right there at the membrane, that's step 5. That's the kickoff, initiating the major downstream signaling cascade.
Exactly. Which brings us to the 2 really big pathways that get triggered right after EGFR activation.
One is the RAS-MAPK pathway. This one is mainly about telling the cell to proliferate, to divide.
And the other major one is the PI3K AKT pathway.
Oh.
This one is super important for cell survival and also cell growth, like the cell physically getting larger.
And you can see why this pathway is such a huge deal in cancer. If you have too many EGFR receptors, or if the receptor gets mutated so it's just stuck on even without a ligand.
Yeah, it just keeps firing off those grow and survive signals constantly.
Super common scenario in lung cancer, colon cancer, head and neck cancers, drives tumor growth.
Which, of course, makes EGFR a prime therapeutic target.
Right. You might use something like cetuximab, a monoclonal antibody that physically blocks the ligand from binding on the outside.
Or you could use small molecule tyrosine kinase inhibitors, TKIs, like erlotinib or gefitinib. These get inside the cell and block the kinase activity itself.
Okay, so let's follow one of those threads downstream from activated EGFR. Let's trace the RAS and mitogen-activated protein kinase pathway. This is where that proliferation signal gets relayed and amplified.
Okay, so you can almost picture this RAS, RAF, MEK, ERK pathway like a bucket brigade. It's passing a signal or a message from the cell surface down into the nucleus.
I like that analogy.
RAS proteins are kind of like the first person in that brigade, standing right there at the inner side of the membrane. They're small GT passes.
Meaning they bind GTP or GDP.
Right, and they act as these critical on-off switches. The switch is on or active when RAS is bound to GTP, and it's off or inactive when bound to GTP.
So how does it get switched on?
Well, when a receptor like EGFR gets activated, it helps recruit proteins called GEFS, guanine nucleoside exchange factors, to the membrane. SOS is one of those.
Right, SOS comes along and basically helps RAS let go of its GDP and grab a GTP instead.
Pop, RAS is now on, active, ready to pass the signal.
And how does it turn back off? It needs to get that GTP back to GDP, right?
It does. RAS itself has a slow intrinsic ability to hydrolyze GTP, but it gets a big speed boost from helper proteins called GTPase activating proteins, aka GAP.
And a really important GAP is neurofibromin 1, or NF1. That's actually a tumor suppressor.
Exactly, because it helps turn RAS off. Now, here's where cancer really loves to step in.
RAS mutations.
Oh yeah, oncogenic RAS mutations are incredibly common. Something like 30% of all cancers, especially big hitters like pancreatic, colon, lung tumors.
And these mutations, they typically mess with RAS's ability to turn itself off.
Right, either they cripple its own GTPase activity or they stop the GTPs, like NF1, from being able to help it.
So the result is the switch gets stuck in the on GTP-bound state.
Constantly shouting, grow, divide, down the line, even when there's no signal from the receptor telling it to.
It just keeps that bucket brigade going nonstop.
Precisely. And one other quick thing about RAS, it needs to be physically attached to the cell membrane to work.
Oh, right, farnesylation.
Yeah, it gets anchored there by adding a lipid group, a farnesyl group, done by an enzyme called farnesyl transferase.
People tried targeting that with farnesyl transferase inhibitors, FTIs, right, as a cancer therapy?
They did. It was an early idea, but it had pretty limited clinical success, unfortunately. Other proteins get farnesylated too, so it wasn't as specific as hoped.
Okay, so back to the bucket brigade. Active GTP-bound RAS is now the signal sender. Who's next in line?
RAS activates the next protein in the chain, which is RAF. RAF is a seronephron kinase. You might also see called MAP3K, MAP kinase. Okay, RAS activates RAF.
And RAF phosphorylates and activates MEK. MEK is MAP kinase, or MAP2K. It's interesting. It's a dual specificity kinase, meaning it can phosphorylate both tyrosine and serotonin residue.
RAF activates MEK, and then MEK activates. MEK activates ERK. ERK stands for extracellular signal regulated kinase, also just called MAP kinase. ERK is kind of the final kinase in this core cascade.
The one who carries the bucket into the nucleus.
That's the one. Activated ERK can actually move from the cytoplasm into the nucleus.
And what does it do in there?
It phosphorylates transcription factors, proteins that control gene expression. Key targets include ELK1 and members of the ETS family.
Phosphorylating them changes their activity.
Right, and alters which genes get turned on or off. The big outcome is turning up genes that promote cell cycle progression, like cyclin D1, which pushes the cell through G1 into S phase. So, proliferation.
But ERK influences other things too.
Yeah, it also plays roles in cell differentiation and survival, depending on the context.
So that whole chain, RAS activates RAF, RAF activates MEK, MEK activates ERK. That's the core RAS-RAF-MEK-ERK pathway.
It's a central highway for proliferation signals. And because RAS mutations are so common and lock that initial switch on, this whole highway is just like frequently jammed and hyperactive in many cancers.
Okay, that makes sense. Now let's switch gears back to the other major pathway that comes off activated receptors like EGFR, or even active RAS itself. That's the PI3K, AKT, and MTR pathway.
Right. And while RAS MAPK is really heavy on proliferation, this pathway is strongly associated with cell survival and cell growth, like physically increasing the cell's mass and size.
So it starts with PI3K, phosphatidylinositol 3 kinase. That's an enzyme that works right at the membrane.
Its job is to phosphorylate a specific lipid molecule that lives in the inner cell membrane. That lipid is called PIP2, phosphatidylinositol 4,5-bisphosphate.
And PI3K adds a third phosphate group to it,
turning it into PIP3, phosphatidylinositol 3,4,5-bisphosphonates.
And PI3K itself, it's usually made of two parts, right? A regulatory subunit, P85, and a catalytic one, P110.
Exactly. And mutations or amplification of the gene for the P110 alpha isoform, PIK3CA, are really common in many cancers, making the enzyme hyperactive.
Okay, so PI3K makes PIP3 at the membrane. What does PIP3 do?
PIP3 is a classic second messenger. It stays stuck in the membrane, but it acts as a critical docking site. It recruits other proteins that have a special domain called a pH domain, plextrin homology domain, which specifically recognizes and binds to PIP3.
So it brings proteins to the membrane. Which ones?
Two key ones. It recruits PTK1, phosphoinositide-dependent kinase 1, and the AKT protein. AKT is also known as protein kinase B, or PKB.
So PIP3 brings PTK1 and AKT together at the membrane.
Precisely. And once they're there together, PTK1 phosphorylates AKT, which is a major step in activating it. Full activation often needs another phosphorylation, typically carried out by a complex called mTOR complex 2, or MPORC2.
So activated AKT, it sounds like a central hub. What does it do?
Oh, it has broad downstream effects. A major one is promoting cell survival.
How does it do that?
It can phosphorylate and inhibit proteins that normally trigger apoptosis, program cell death, things like BAD or GSK3 beta. It can also lead to the activation of anti-apoptotic factors like NF-kappaB, which we'll get to.
So actively stops the cell from dying. What else?
Another huge role is promoting cell growth and proliferation. Active AKT does this largely by activating another complex called mTOR complex 1, or mTORC1.
mTORC1, that stimulates protein synthesis.
Yes, through downstream targets like S6K and 4E-BP1. It also promotes lipid synthesis. Basically, it drives the cell to get bigger and helps it commit to dividing.
Okay, so this PI3K KD-mTOR pathway is clearly powerful for growth and survival. It must have a brake, right?
Absolutely essential. And the critical tumor suppressor brake on this pathway is PTEN, PTEN phosphatase, and Kensin homolog.
PTN. It's a phosphatase, so it removes phosphates, but it works on lipids.
Exactly. It's a lipid phosphatase. Its specific job is to dephosphorylate PIP3. It removes that third phosphate, converting it back into PIP2.
Ah, so it directly reverses the action of PI3K.
Precisely. It shuts down the signal right at the source, preventing AKT recruitment and activation. And loss of PTN function through mutation or deletion is incredibly common in a huge number of cancers.
So if you lose PTN,
you get unchecked hyperactivation of the entire PI3K-KT-mTOR pathway. Constant signals for growth and survival. It's one of the most frequently lost tumor suppressors in human cancer.
Wow. Okay. Trying to keep track of that membrane action. PI3K makes PIP3 from PIP2. PIP3 recruits PTK1 and AKT.
Remember the mnemonic? Pipes 3 picked AKT. Helps remember that PIP3 brings PTK1 and AKT to the membrane. PIP3, PTK1, AKT.
Pipes 3 picked AKT. Got it. Okay, let's move on to another huge player.
Myc, often called the master conductor or maybe orchestrator of proliferation and a lot more.
So Myc, referring to the family members Myc and NMyc and LMyc, these are powerful proto-oncogenes. They encode transcription factors, right?
Right, meaning they bind directly to DNA and control the expression of a whole bunch of other genes. Myc binds to specific DNA sequences called EBOX sequences.
And it regulates a massive number of genes, right? Like 10 to 15% of the whole genome.
It's incredible. Because it controls so many genes, Myc has really diverse functions. Number one is promoting cell proliferation. It cranks up genes that drive cell cycle entry and progression, things like cyclins and CDKs, while at the same time, it turns down inhibitors like P21 and P27. Hits it from both sides.
It also drives cell growth, making the cell physically bigger.
Yeah, it stimulates things like ribosome biogenesis, protein synthesis. It also influences cellular metabolism, how cells use glucose and glutamine.
Interesting. I also heard it has a kind of paradoxical role in apoptosis.
It does. Myc can actually make cells more sensitive to programmed cell death, especially if they're under stress.
But in cancer, that effect gets overridden.
Often, yes. Usually by other co-occurring changes, like if the cell is also overexpressing anti-apoptotic proteins like BCL2. Myc also contributes to making cells immortal, partly by upregulating telomerase.
And Myc expression itself is tightly regulated?
Normally, yes, by those same growth factor pathways we've been discussing, like RAS and PK and WNT signaling.
But, like everything else we've discussed, it gets dysregulated in cancer.
Very commonly. Through various mechanisms. Gene amplification, making many copies of the Myc gene. Yeah. Chromosomal translocations.
Like in Burkitt's lymphoma.
The classic example, yeah.
Yeah.
Where the Myc gene gets moved next to really active immunoglobulin gene enhancers, driving massive overexpression. Or Myc can be activated just because upstream pathways like RAS or PI3K are constantly firing.
And there's that classic cooperation between Myc and RAS.
Yeah, it's a cornerstone of cancer biology.
Yeah.
Neither oncogene alone might be enough to fully transform a normal primary cell into a cancer cell.
Yeah.
But if you have both active cytoplasmic RAS signaling and hyperactive nuclear Myc,
then you get robust transformation.
Exactly. It really underscored that multi-hit nature of cancer development. You usually need multiple pathways to go wrong.
Okay, one more major pathway to cover, NF-kappaB.
NF-kappaB, the pro-survival and pro-inflammatory hub. And this one is particularly relevant for us in radiation oncology.
Why is that?
Because radiation itself can activate it.
Yeah. Okay. So NF-kappaB stands for nuclear factor kappa light chain enhancer of activated B cells. Bit of a mouthful.
It is, but basically it's a family of transcription factors. A common active form is a dimer, often the P50 and ReLA, which is also called P65, subunits together.
And it's involved in immune responses, inflammation.
And critically for cancer, cell survival, and proliferation.
So how does the main or canonical NF-kappaB pathway get switched on? Where is it usually?
In its normal inactive state, NF-kappaB is held captive in the cytoplasm. It's bound by inhibitory proteins called I-kappaB proteins. The main one is I-kappaB alpha.
And I-kappaB alpha holds onto it.
And masks its nuclear localization signal, or NLS. So NF-kappaB physically can't get into the nucleus where it needs to be to turn on genes.
Okay, so what triggers its release?
A whole wide range of stimuli. Pro-inflammatory cytokines like TNF-alpha and IL-1, bacterial bits like LPS, viral infections, oxidative stress.
And you mentioned genotoxic stress.
Yes, which includes DNA damage caused by radiation. That's a key activator.
So these stimuli, what do they activate upstream?
They activate a critical complex called the IKK complex, I-kappaB kinase complex. It has catalytic subunits, IKK alpha and IKK beta, and a regulatory subunit called NEMO, or IKK gamma.
IKK gets activated. What does it do?
Its main job is to phosphorylate I-kappaB alpha on specific serine residue.
And that phosphorylation is like a signal.
Exactly. It's a tag marking I-kappaB for destruction. It's recognized by an E3 ubiquitin ligase,
which adds ubiquitin chains.
Targeting I-kappaB for degradation by the proteasome, the cell's garbage disposal.
So I-kappaB gets chewed up. What happens to NF-kappaB?
It's now free. Its nuclear localization signal is unmasked. So NF-kappaB can translocate, move into the nucleus.
And once it's in the nucleus,
it binds to specific DNA sequences called kappa B sites. These are found in the promoter or enhancer regions of its target genes. Binding there activates transcription.
And what kinds of genes does NF-kappaB turn on?
A whole host of genes that drive pro-cancer activities. Things involved in inflammation, immune response modulation, but crucially, cell survival.
How does it promote survival?
By turning on potent anti-apoptotic proteins. Think BCL2, BCLXL, CIAPs, FLFP. These directly block cell death pathways. This is really critical for treatment resistance.
What else?
It can also promote proliferation by turning on cyclin D1, stimulate angiogenesis by upregulating VEGF, and even promote invasion and metastasis by turning on MMPs, matrix metalloproteinosis.
So in cancer, NF-kappaB is often just stuck on.
Frequently, yes, constitutively active, constantly promoting survival, resistance, inflammation, and growth within the tumor microenvironment.
And the fact that radiation itself activates it, that sounds like a problem.
It can be a double-edged sword. While NF-kappaB activation is part of the normal inflammatory response to damage, in cancer cells, it can switch on those pro-survival genes, potentially helping the tumor cells survive the radiation hit, makes them more radioresistant.
Which is why people are looking at inhibiting NF-kappaB.
Exactly. As a potential strategy to make cancer cells more sensitive to both chemotherapy and radiation.
You know, thinking about how all these pathways get tangled up, EGFR feeding into RasMAPK and PI3K and how things like NF-kappaB contribute to survival. It really helps frame how our targeted therapies work, or sometimes why they don't work if there's resistance.
Absolutely. Let's think about a mini case. Consider a patient with, say, locally advanced head and neck cancer. They're getting radiation.
Okay.
If we know their tumor has high expression of EGFR, maybe from immunohistochemistry on the biopsy,
Right.
We know from clinical trials and understanding this pathway that adding an EGFR inhibitor, like cetuximab, along with the radiation can actually improve outcomes.
So that's a direct clinical application. We're targeting that specific antenna, the EGFR, that's driving that tumor's growth to hopefully make our radiation work better.
Precisely. It shows how understanding this molecular wiring informs our actual treatment decisions.
Okay, that was a lot of complex biology. Let's try to pull out some essential clinical pearls to really lock this in for boards and for practice.
Good idea. Pearl number 1, EGFR. It's a key target, especially in epithelial cancers. Lung, colon, head, and neck are big ones. And inhibitors like cetuximab or the TKIs like gefitinib, erlotinib, they're used clinically, sometimes combined right with radiation.
Okay. Pearl 2, RAS mutations. Super common, remember that 30% figure. And critically, having a RAS mutation downstream can affect whether a patient responds to an EGFR inhibitor upstream.
Yeah, if RAS is stuck on anyway, blocking EGFR might not help much. Directly targeting mutant RAS itself has been really tough historically.
But targeting things downstream, like Myc inhibitors, that's showing more promise now, right?
Definitely. That's an active area. Okay, Pearl 3, the PI3K AKT-mTOR pathway.
Hugely important. Frequently hyperactivated in countless cancers, often due to those PIK3CA mutations or, very commonly, loss of the PTN tumor suppressor.
And this pathway heavily influences survival and growth, and also radiosensitivity. Inhibitors targeting different points PI3K itself, AKT-mTOR, are either approved or in late-stage development for various cancers.
Pearl 4. MYC, that master regulator transcription factor. It's powerful, commonly dysregulated, controls a huge gene network for proliferation and growth.
And remember, it's cooperation with pathways like RAS, while MYC itself is notoriously hard to target directly with drugs.
Its status, like amplification, can definitely tell you something about how aggressive a tumor might be and potentially how it responds to treatment.
Right. And finally, Pearl 5, NF-kappaB.
The crucial pro-survival factor, often constitutively active in cancer, driving resistance, including radioresistance.
And don't forget, radiation itself activates it. That's key. So targeting NF-kappaB is being actively explored as a way to sensitize tumors to radiation and chemo.
Okay, those are great takeaways. Ready for a quick board blitz? Let's see how these concepts might pop up in a multiple choice format.
All right, let's do it.
Question 1. Ligand binding to the epidermal growth factor receptor, or EGFR, typically initiates signaling by first causing A. Internalization of the receptor, B. Activation of RAS GT-PACE activity, C. Receptor dimerization and autophosphorylation, or D. Nuclear translocation of EGFR.
Okay, first step after ligand binding has to be C, right? Dimerization brings the kinase domains together for autophosphorylation.
Exactly. C is correct. The dimerization and phosphorylation are the immediate consequences that then recruit everything else.
Question 2. A point mutation in a RAS proto-oncogene that leads to its constitutive activation typically impairs which of its biochemical activities? A. Binding to GDP, B. Interaction with guanine nucleotide exchange factors, GEFs, C. Farnesylation, or D. Intrinsic GT-PACE activity, or GA. P-mediated GTP hydrolysis.
Right. The key problem with oncogenic RAS is it can't turn off, so that points to D. It impairs its ability to hydrolyze GTP back to GDP, either its own slow activity or the help it gets from GAP.
Perfect. Answer is D. It gets stuck in the GTP-bound state.
Question 3. The tumor suppressor, PTEN, antagonizes the PI3K and AKT signaling pathway primarily by A. Directly phosphorylating and inactivating AKT. B. Preventing PI3K from binding to activated growth factor receptors. C. Dephosphorylating the lipid second messenger PIP3. Or D. Sequestering PDK1 in the cytoplasm.
PTEN. That's the PIP3 phosphatase. So it has to be C. It removes the phosphate from PIP3, converting it back to PIP2 and shutting down the signal.
Correct again. C is the answer. It directly counteracts PI3K's action.
And question 4. The Myc oncogene primarily functions as A. Receptor tyrosine kinase, B. Cytoplasmic serotonin kinase, C. G protein coupled receptor, or D. Nuclear transcription factor.
Myc, this one should be fairly straightforward. It binds DNA, controls genes. It's D, a nuclear transcription factor.
Absolutely, D is correct. Classic transcription factor. Yeah. Okay, last one. Question 5. In the canonical NF-kappaB signaling pathway, the IKK complex is activated by upstream stimuli. What is the immediate downstream consequence of IKK activation? A. NF-kappaB translocates to the nucleus. B. I-kappaB binds more tightly to NF-kappaB. C. I-kappaB is phosphorylated and subsequently ubiquitinated and degraded. Or D. NF-kappaB directly phosphorylates target gene promoters.
Okay, IKK gets activated. Its immediate job is to phosphorylate I-kappaB. That phosphorylation then leads to degradation, so the immediate downstream consequence is C.
Exactly. C is the right answer. IKK phosphorylates I-kappaB, which then leads to its degradation. Then NF-kappaB goes to the nucleus. C is the immediate step.
2. Okay, so let's try to quickly recap this whirlwind tour of cancer cell wiring.
We started with EGFR, that cell surface antenna, triggering 2 big pathways downstream, The RAS MAPK mainly for proliferation.
And PI3K mTOR really pushing survival and growth, with the crucial P10 acting as the brake on that second one. We looked at RAS proteins as those key molecular switches needing that farnesyl anchor at the membrane, and how they're so often mutated to be stuck on, constantly driving the RAS-MEK-ERK cascade.
Then we hit MEK, that powerful nuclear transcription factor like a master conductor, cooperating with pathways like RAS, controlling proliferation, growth, metabolism, and more.
Finally, NF-kappaB, held inactive by I-kappaB, but unleashed by the IKK complex in response to all sorts of stimuli, including radiation itself.
Right, and once unleashed, it promotes a ton of pro-cancer activities: survival, anti-apoptosis, inflammation, proliferation, angiogenesis, you name it.
So understanding how these pathways get dysregulated, how they crosstalk, it's just fundamental to understanding cancer biology and, importantly, identifying targets for therapy, including ways to potentially make radiation work better.
It definitely feels like a lot to keep straight, I know. Sometimes after reviewing all this, my brain just feels like alphabet soup with all the acronyms floating around: EGFR, PI3K, MAPK, EK, AKT, IKK, NF-kappaB.
Huh. Yeah, it's like trying to remember everyone's name tag after a massive conference 3 months later. Did I really learn anything or just collect acronyms?
Well, the key is just keep revisiting these core concepts, connecting them together, and eventually it really does start to click and make sense in a clinical context.
Absolutely. And you can definitely solidify this knowledge and test yourself. Complete practice oral boards over at radoncSmartLearn.com.
Right. And be sure to subscribe to RadOnc Smart Review for our next exploration. We'll actually build directly on these pathway concepts when we discuss the hallmarks of cancer. Thanks everyone for tuning in.